right. So we're going to get started a little bit here. I think people are still rolling in a little bit, a little stragglers, but we can get started here because I know that Aaron has a lot to talk about today. And so I want to be able to give him as much time as possible so that we can get to our Q&A section. So let me just quickly introduce Aaron, or maybe I should say I am Rob Corteau. I'm second vice president of NAMA. I'm just the host today. Aaron's the special guest of honor here. So that's all I'm going to say about me. So Aaron is a mycologist, an educator, a community organizer, as well as the founder of the Mycoverse, to which I will link in the chat. Aaron is the current chair of NAMA's Conservation and Stewardship Committee, to which I'll also share a link. And to add to an already impressive resume, Aaron also does work with the Fungal Diversity Survey, predominantly as a fungi collector, I believe. Last but not least, Aaron is based in Los Angeles. And I saw from the chat that there are a few of you also from Los Angeles and at least from the greater California. I would like to express my appreciation to Aaron for taking the time to give this webinar. As we all know, Los Angeles has been struck by a pretty horrific fire. And so if anyone here wishes to donate to the relief efforts, I'll also be posting a little link to that in our chat. Since this is a free webinar, perhaps you can contribute just a small amount to help out the folks out there. And that would be very much appreciated. So thank you in advance for that. I'd also like to remind everyone that they can post questions in the chat, but even better is to post your questions in the little Q&A section. There's a little button at the bottom of your screen below the video, just says Q&A. That's the greatest spot to post your questions because it just saves them until we're ready to answer them and we don't get them lost in the chat. So please use the Q&A function if possible. If that's too confusing in one way or another, maybe you are on a cell phone and can't find it pretty easily, you can post into the chat and I'll try to record it quickly. So without further ado, Aaron, I'm handing it over to you.
All right. Thank you, Robert, for hosting tonight's talk, and thank you, NAMA, for having me tonight. And yeah, I'm going to jump right into it. So let me introduce myself. Thanks for that lovely introduction. Again, my name is Aaron Tupac, and I use they, them pronouns. I am currently evacuated to the Mojave Desert on Western Shoshone and Serrano land outside of Los Angeles, whose land is Tongva Ganglino, where I live. And Robert, let me know if you can't see my presentation. I think we can.
Yeah, looks good.
Great. I'd like to start off this evening taking a moment of silence and reflection on the monumental devastation of the ongoing winter fires in Los Angeles, the worst in the city's history for the many who lost their homes, human and more than human. This is a picture of me in Eaton Canyon. The fires that continue to burn in Los Angeles have devastated my community and me here in Los Angeles. It is overwhelming as I try to wrap my head around things. The lives of so many have changed so quickly, human and more than human. I am grieving the loss of so many lives, including the fungi and plants I love so dearly. I haven't truly begun processing the impact these fires have had. My livelihood has been based in these forests and urban areas that have burned. I fell in love with fungi and found my community in these places. These areas where I do my fungal conservation research and lead fungi education classes, and they are filled with the places I love. They are where I go to rest and recreate in the big city. Eaton Canyon in particular is a special place for me. I wrote about it in my upcoming book about mushrooms and fungal conservation. I met my wonderful romantic partner, Vanessa, on a mushroom walk I was leading in the canyon with the California Native Plant Society, on just one of the many mushroom walks I organized there. My partner grew up a few blocks away from the canyon, and we've spent our special occasions, birthday celebrations, and holidays in these woodlands, finding joy and wonder. The fires are a timely topic, as one of today's main topics looming in the background of my talk is climate change and how we can actively take measures to counter and effectively slow down climate change, how we can change our relationship with the land. I recently heard something profoundly insightful, and I would like to share and frame this talk around tonight. The Potawatomi botanist and indigenous wisdom keeper, Robin Kimmer, during a book talk of her upcoming book, during a Q&A, she was asked about what we can do about the overwhelming calamity of climate change. She responded by saying, "Let grief stop us in our tracks." Just as she shares in her bestseller book, Braiding Sweetgrass, Indigenous Wisdom, Scientific Knowledge, and the Teachings of Plants, if grief can be a doorway to love, then let us all weep for the world we are breaking apart so we can love it back to wholeness again. I take this as an invitation to feel the hard feelings, reflect, and notice how the human world responds. We need to stay with the trouble, be kind to ourselves, and feel through this. So, where we're going through tonight's talk, let's go over. I want to share the outline so you all know the order of events for tonight. So, to begin with, I will give a little background into my story, where I'm coming from, and how I got into conservation mycology. And the following will be the central questions I will entertain during tonight's talk. I'm hoping to provide some insight to anyone interested in creating better relationships with fungi that I had wished I'd known when I started my journey into the world of conservation. So first off, how conservation mycology is directly responding to climate disruptions and habitat destruction. What is conservation mycology and how is stewardship a key part of it? Where we can begin with fungal conservation and stewardship, how you and your community and fellow NAMA members can get involved. And ultimately, how can fungi help us inspire and rethink conservation? Where do we see the future of conservation and stewardship going? And to conclude our time together, I will share an excerpt from my upcoming book on fungi and conservation from a chapter I'd previously written about how I fell in love with fungi in Eden Canyon, to end on a brighter note. Then hopefully there'll be some time, as Robert said, for some Q&A where I can take a few questions. All right, let's begin. So during the darkest time of my life, amidst the COVID-19 pandemic, falling in love with fungi saved my life. When the world felt like it was falling apart, when loneliness and chaos felt at their peak, I started going on walks looking for fungi. To my surprise, I found joy and wonder in the big city of Los Angeles through encountering many remarkable fungi and many wonderful, inspiring people along the way. I had no idea how much joy and meaning a simple encounter with a mushroom would bring. Going out to meet mushrooms during this time was a remedy for when I felt alone and isolated, as I was thousands away from my family in Minnesota, where I grew up. As a transplant, I didn't have a strong community at the onset of the pandemic. The overwhelming grief of it all stopped me in my tracks. Grief invited me to slow down and reflect on what matters most to me. What made life worth living? What makes me feel alive and inspires me? Who did I want to surround myself with? Then, in 2020, the book Entangled Life, How Fungi Make Our Worlds, Change Our Minds, and Shape Our Futures by Merlin Childerick came out. which filled me with that sense of aliveness and childlike wonder, learning about the lives of fungi. The book changed my world. It inspired me to start noticing fungi. I soon became strikingly aware of my dependence on and relationships with the overlooked kingdom of fungi. After becoming aware of these interconnections, I couldn't unsee them. Learning about fungi helped me recall the joy and healing powers of noticing the natural world, where I had learned growing up in Minnesota the healing and inspiring power of going for a walk in the woods. I immersed myself in the world of fungi, traveling near and far to meet mushrooms and fungal enthusiasts. Soon after dedicating my life to fungi, I was hired for my first job in fungal conservation. Through the first statewide initiative to document fungi, the California Fungal Diversity Survey Project, I became a paid and employed fungal community scientist for the nonprofit, the Fungal Diversity Survey, also known as Fundus. This is the first time the government has sponsored a project like this, where our primary funders are the state of California and California Institute for Biodiversity. To my surprise, this baseline work had never been done before. We know the native plants of California, but we don't know the native fungi of California. While there has been a lot of excitement around documenting and describing new fungi, I couldn't help but think about what were we doing for fungi? What efforts were there for protecting and helping support fungi? This led me to delve even further into conservation in 2023, where I helped found the committee dedicated to conservation and stewardship here at the North American Mycological Association. I've been the chair since the inception, creating an international coalition between conservation organizations from around the world and continent, leading literature reviews of all the research out there, compiling educational resources, which I'm excited to share about in a bit, and much more I'm excited to share with you about later in this talk. In addition to being an organizer abroad, I am also a community organizer locally, and I am based in Los Angeles, where I found the values-based fungi enthusiast organization called Exploring the Mycoverse, currently hosted at Arlington Garden in Pasadena, California. Since its founding in 2021, during the pandemic, Exploring the Mycoverse has had over 100 events in the first 3 years, engaging over 1,000 people, making fungi education free and open to the public. We're a budding group of community scientists, artists, naturalists, and so on. Falling in love with fungi helped me reclaim responsibility, meaningful connection, and hope. Fungi taught me how to reach out, to myceliate in all directions, how to connect through difference. Fungi inspired me to go out of my comfort zone and create the community I needed. So, getting into the meat of the talk, how conservation mycology is directly responding to climate disruptions and habitat destructions. The threats facing fungi are largely the same as those confronting other species. The IUCN ranks the following threats to fungi by severity: land development, agriculture, logging, climate change and severe weather, fire and fire suppression, invasive species, pollution, disturbance, transport and service corridors, energy production and mining, collecting, and water management and usage. All of these, many of the threats listed above, are different ways in which the habitats of fungi are destroyed. Habitat loss is the primary cause of biodiversity decline around the world. Habitat destruction dramatically alters fungal communities due to changes in local microclimates, loss of plant partners, and increased soil erosion. to name a few. Also, a lot of these are related to agriculture expansion, aggressive tillage, extensive chemical fertilizers, fungicides, and pesticides destroy underground fungal networks. Without their fungal partners, crops need more chemical inputs and are more susceptible to drought, or soil erosion, pests, and pathogens. And lastly, these are all related to some extent to climate disruption. Extreme temperatures, drought, and floods threaten mycorrhizal fungi's ability to transport nutrients and sequester carbon. Disruptions from climate change, such as intense wildfires, destroy plants and the underground mycorrhizal networks. So, conservation mycology is rooted in responding to these threats. Yet, fungal conservation is in its infancy. Despite fungi's enormous contributions to the shaping of our ecosystem, it is estimated that more than 90% of fungal species remain unknown to science. Conservation mycology today is summed up in only a handful of peer-reviewed papers published on fungal conservation, stewardship, and best practices. There are very few paid positions in conservation-focused mycology. There are very few academic programs, and there are very few resources dedicated to this field yet. There are a handful of organizations dedicated to conservation, to the conservation of fungi. A lot of people don't know where to begin with conservation mycology. So my hope is this talk gives us a place to begin and gives us some ideas on how we can organize and be intentional on focusing and growing fungal conservation from both top-down approaches and bottom-up approaches, as we need both. We need all hands on deck, so to speak. So, what is conservation mycology today? Kew Gardens in London helpfully shares a report every few years about the state of conservation mycology that is primarily focused on the state of fungal collection efforts. I recommend checking out the Kew Gardens website or our Conservation and Stewardship resources page, where we share the link to the latest report. Though in this talk, I will be utilizing a paper I found foundational to the field that illustrates the large complexity and many strands of conservation mycology. Published in 2018 in the Journal of Conservation Biology, recognition of the discipline of conservation mycology was written by many familiar mycologists you may recognize. Tom May of Victoria, Australia's Botanic Gardens. We've got Jerry Cooper, Swedish mycologist Anders Dahlberg, Juliana Furci of the Fungi Foundation, David Minter, Greg Mueller of the Chicago Botanic Garden and NAMA member, Australian mycologist Alison Pouliot, author of Meetings with Remarkable Mushrooms that the NAMA book club just read together a few months ago, that fellow Conservation and Stewardship Committee member Georgie Voros led, and Zhuliang Yang of the Chinese Academy of Science. So they outline some really helpful characteristics which make fungi uniquely difficult and challenging to conserve. Before we get into the research agenda, I think these characteristics are really helpful to understand where it's conservation mycology is coming from. So first off, diversity. There are likely about 3 million fungal species globally. More than 90% of the species have yet to be named. We're dealing with invisibility. The spores, mycelia, and many sporing bodies are microscopic and not visible to the naked eye. They spend most of their lives out of sight underground. Sporing bodies, mushrooms, are often ephemeral, lasting for short periods of time, making them difficult to encounter, observe, collect, or study. And they're also overlooked. Fungi are largely absent from conservation policies and management. They're also absent from nature education in curriculums and educational interpretations in natural areas, like signs and booklets at parks. Then reference collections. We have limited collections of samples of fungi, far from representative of the diversity of the fungal kingdom. We're just starting to create a comprehensive DNA barcode library, which we can use for interpreting environmental DNA. For instance, if you attended Serena Whitner's talk, you may have heard her talk about how marine mycology work only mapped to terrestrial fungi or fungi found on land. Because, for instance, because we only know so many terrestrial fungi, and marine mycology does not have any conservation efforts yet that we are aware of. And for reference collections, we're still in the beginning of creating a fungal tree of life for studying and understanding the evolution of fungi. It is far from comprehensive and is currently still changing frequently due to the advancement in molecular analysis technology. Then we have scale. As many of you know, some fungi are extremely tiny, like microscopic inside, like the ones growing and living inside insect guts or endophytes and leaves, or this one like crusts, growing on decomposing litter, to really large, to covering landscapes, growing to be among the largest organisms on the planet, like armillaria and the humongous funguses of Michigan and Oregon, and many more that we have yet to get to know. Then we encounter the challenges of language. So individuals or fungi are their own kingdom of life, separate from the natural world, albeit often being lumped under plants or microbes, which hides their biological and ecological functional uniqueness. We use words like fruiting body and vegetative body instead of sporing body and mycelia, or even we use, say, colonizing instead of myceliating. A lot of issues with language. Then we have cultural attitudes. As many fungi lovers have encountered, the fear of fungi. In many of our cultures in North America, fungi are often considered or regarded as pests rather than integral components of our ecosystems. As many we saw recently, it manifested through the hit TV series The Last of Us that has made a massive impact on how people understand and view fungi. And then we have the challenges of interconnectedness. Their entangled way of living with other organisms. Most fungi are symbionts, making fungi difficult to test cause and effects through traditional ways of biological studies of isolating species to test. Also ecological studies often deal with fungi and their partners separately instead of one living entity or holobiont like these lichens. Then we're facing changes in paradigms. So current conservation assessment paradigms use traditional single species approaches, such as via red listing. These efforts are still in their infancy, with only a few countries in the world having them, North America being largely absent with only a few red listed lichens. But guild and whole community conservation assessment approaches are also required due to factors like their interconnectedness, requiring a shift in paradigms to system and community conservation assessments too. Lastly, we have the challenge of communication in compiling existing information that's out there about fungi conservation and translating it and sharing it between researchers, land managers, policymakers, and community scientists and the public, like you'll hear tonight. All right, here's those are like the 10 main characteristics and challenges that fungi face. And then we have our research questions that are based on these. So how do we address these issues? What questions are conservation mycology asking? How do we use ecological knowledge for the 10% of the fungi that have been described and named by science to effectively conserve all fungi? An ecological focus on fungi, plant partnerships, complete carbon cycles, inclusive of decomposition, climate disruption, and so on. Can we monitor and use effective surrogates or proxies to create fungal conservation reserves? So for example, here in Southern California, maybe good candidates for surrogates would be, for plants, would be live oaks for oak woodlands or chamise for chaparrals habitats. Can we make the assumption that if live oaks are doing well, are their associated fungi doing well? For an example, another example, if our canyon live oaks, very specific to SoCal, if our canyon live oaks are doing well, maybe then we can assume our oak associates are doing well, such as the Helvella Dryophila group, or maybe our Lactaria silanicola, if they're doing well, the golden milk caps, who are known to grow in older woodlands, then perhaps the health of our forests and other fungi are doing well as well. How does the grain and patchiness of fungi distributions affect these conservation efforts via surrogates? And how do we standardize the monitoring of fungi so we can use it in government regulations and policy through required surveys before developing land, such as through environmental impact reports? It is really important that we get fungi included in environmental impact reports, but we need to show how we can do that first. I know here at Fundus, when we started our CA Fundus project, we had to come up with our own methodology on what data would be most helpful and plausible to collect. But there's no standardization in place yet, leading community science observations to tend to be a bit disorganized or inconsistent, making them hard to analyze and make sense of. And then how do we best detect and monitor rare fungi through the collection of sporing bodies and DNA sequencing of soil samples, what's also known as metabarcoding? And how do we detect their absences? How do we know how rarely occurring fungi are doing? How do we integrate both of these approaches to monitoring of DNA sequencing of soil and collection of sporing bodies? And it is something we have been trying to figure out at Fundus by working with organizations like the University of California's Cal eDNA program, who has been performing biodiversity sequences, DNA sequences of the soils here. But what they're able to detect in the soils might be the presence of a fungus, but that doesn't necessarily mean that the fungi are alive. So we are working to coordinate with them to go visit those sites to see if we can collect sporing bodies during potentially ideal mushrooming conditions. What are better ways to to detect if a fungus is alive. And then we have, what are effective stewardship practices for fungi? And how do these practices compare to existing stewardship practices designed for other habitats, plants, and animals, especially in relation to fires? How does fire affect fungi? And how can we help fungi after fires? Or for agriculture, or for silviculture, managing forests of timber, or grazing regimes, just as a few examples. And then how do life history characteristics vary across fungi? Their different lineages and guilds. How do we measure these? So how often do they come up? What do they look like? There's a lot of variation. And how do we challenge and mitigate negative um oh actually do and then how do we challenge and mitigate negative attitudes about fungi among the public, land managers, and policy developers? As I mentioned, mycophobia before, the as fungi have been stuck in the death, disease, decay categories. And how do we engage foragers in conservation? So collecting and eating fungi are a major point of contact with fungi in many of our cultures. So how do we encourage a reciprocal relationship and not just a relationship based on consumption or what a fungus can do for you? So, all right. Now that we've acquainted with what questions conservation mycology is currently pursuing, let's take a look into who's doing fungal conservation and some examples of fungal conservation in action that I particularly like. And here's just a list of all of the questions. I don't want to list them out right away so it's not all overwhelming, but that is the very large field of conservation mycology. A lot of different facets. So who's doing fungal conservation? We've got the Fungi Foundation, the Fungal Diversity Survey, we've got the IUCN Committee, we've got SPUN, the Society of Protection of Underground Networks, Kew Gardens, just to name a few. First, I'm going to share about a little bit more about the CA Fundus project I'm a part of due to its exciting nature that I think that can be modeled for how other programs can form and get funding. It's been an honor working as a paid community science to collect and document the fungi of LA, bringing me to survey the mushrooms of Los Angeles, their mountains, backyards, canyons, gardens, beaches, chaparral, forests, and deserts, all over the place. And as I had previously mentioned too, this project is the first US statewide project of its kind. For the first time in California history, the California State Legislature has invested in documenting the fungi of California. The CA study, this is part of the California state government's commitment to preserving 30% of lands and waters by 2030, which is also known as the 30 by 30 initiative, which is something very on a larger scale, even internationally, that countries, states, regions are working towards meeting. And through the California government's commitment to do the 30 by 30 project, they have made funding available for biodiversity research. So unprecedentedly, fungi were included in 30 by 30 goals, allowing us to get funding to document fungi. So As of 2024, the CA Fundus team has made over 10,000 collections over the course of 2 years. Our team is a small group of passionate community scientists, well over 10 fungi collectors and over 5 DNA sequence validators, and a bunch of other staff that helps us make this happen. The fungi collectors, that's my job, go out into the field and are tasked to collect and document detailed data about fungi, particularly about their environmental associations and relationships. We take detailed, high-quality photographs of mushrooms themselves and the environment we find them in. We take detailed notes on various aspects such as smell, the substrate they're growing on, and UV 365 nanometer reactions, chemical tests like KOH reactions. And yeah, I think perhaps the most interesting of all is that is the least amount of study is taking note of what plants they are growing with and possibly associated with. So when we're out there, fungi collectors collect a few of the fresh mushrooms or fungi in the field to dehydrate and send off to our DNA sequencing team and our fungariums at universities for future study. The CA Fundus project makes the DNA sequences, results, and fungarium collections available to the public and scientists around the world to study the mushroom collections further. During the first year of the project alone, we found that 20% of the DNA sequences collected do not match any other sequences in publicly available databases, meaning these fungi have yet to be documented in such a way. In Southern California alone, I have encountered abundance of species who have not yet been documented here. The CA Fundus project is only just an example of a small group of passionate community scientists. The fungi also need you to help document their appearances, life histories, and relationships too. I wanted to share a little story about my favorite part, one of my favorite stories working on this project, and the advantage of working as a community scientist has allowed me to study things in detail that traditional institutions haven't had a chance to. One of the best examples I have is of the time in August 2023, when the unexpected happened, when Southern California received its first ever tropical storm warning, which in August is usually our historic fire season, not January, when it's typically raining, our wettest season. So Hurricane Hillary brought incredibly rare summer rains, and as fungi lovers know, rains bring mushrooms. And we mobilized quickly, got my friends together, colleagues together, and we went out and found a lot of really interesting mushrooms, encountering undescribed Lactarius around the size of our heads. Like, I think I've got one somewhere here of Dean. Just massive, really interesting. And then the beautiful blue cyanula on our right. Tiny orange clusters of Xeromphalina, cirrus on the top left of your screen, and peculiar little orange disc shaped mushrooms that I found really interesting that smell awfully like sewer gas that turned out to be Geopixus remii. During this period, we made some of the most significant contributions to the community science to our project. So rarely, if ever, had these been documented, and if they had been documented, definitely not during this time of year. Some of which had been thought to be snow melt fungi, like Chromacera here, but doesn't appear to be the case. It is thought that a lot of these mushrooms that we had the honor of documenting during this time only come up every several decades when the right conditions and rains come their way. Another rare opportunity awaits us as community scientists here in LA while we get ready to survey the burn scars of the Eaton fire and the Palisades fire after we receive rains in the winter to study fungi's relationships with fire. All right. I want to share a bit about, oops, there we go. Our Conservation Committee. This is another example of where we're trying to do conservation here. So what we've done as a committee since our founding in the summer of 2023, we have worked with over 30 NAMA members to put together the committee to give us the direction and assistance. I thank all those who showed up and helped out and sat through those meetings and contributed. It's been an incredible learning experience. In our first year, we led a literature review of the main conservation mycological publications. And then in our second year, we created a web page listing key resources from that review where you can learn more about fungal conservation. And we tried our best to succinctly summarize what we had learned. We put a lot of work into it. I'm proud as nothing like this quite existed before. And it was something I wish I had when I was getting into fungal conservation. I want to make a special thanks to our Conservation and Stewardship Committee members for helping out putting this together. Amy Honan, Dina Tempest Thomas, Glenn Rogers, Greg Wolf, Georgie Voros, Hannah Huber, Nora Dunkirk, Bryth Eugene, Tay Bright for generously helping volunteering their time and energy. Another thing I'm proud that we've done is connect with fungal conservation groups like Society of Protection of Underground Networks, the Fungi Foundation, the British Mycological Society, the Natural Heritage Programs across the states, such as in Oregon and Pennsylvania, and the Fungal Diversity Survey. And I will share more in a bit how you can get involved in our committee and the future of our committee. We're actively looking for members to come join us and learn more. You can, if you're an AMO member, you can join our meetings. And then another group organization doing really remarkable fungal conservation that I'm really a big fan of is the Fungi Foundation. They released a fungal conservation tracker last year, and they host an elders program gathering anthropological and cultural and indigenous usages in relationships with fungi. They're working on a fungi education curriculum that's released. It's free to the public on their website for kids. It's really good. I use it a lot. And they work with artists, film producers, and museums to get fungi in public spaces. And last year, they got the governments of Chile and the UK to make a fungal conservation pledge with the aim to incorporate fungi into national and international legislation, policies, and agreements. And there's a few other countries that signed on to this pledge, two North American countries, Mexico and Costa Rica, and as well as Colombia, Spain, Peru, Ecuador, Cambodia, Guinea, Benin, and Germany. And Fungi Foundation is doing so much more. I highly recommend checking that if you have not. So, how can fungi inspire us to rethink conservation? Where do we think the future of conservation mycology and stewardship is going? Yes, monitoring and sequencing is essential, but also community organizing, noticing Grieving and loving are essential process to the process too. They are part of a larger process of conservation. So noticing without direct action for the sake of beauty and awe to inspire us to love the world back together, as Ronald Kimmer reminded us. One of the examples that I think that really can change conservation is values-based community organizing with an intentional focus on building better relationships with fungi. Another that we practice, I teach a lot, is for conservation by exercising our attention of fungi, becoming more fungally aware, getting to know and notice fungi, where noticing their life cycles, their life habitats, their habits, their life habits, their habitats, who they're associated with and connected to or growing on or near. I really like how the poet Mary Oliver said, attention is a form of prayer if it isn't about making money. I think you could say if our attention isn't focused on what fungi can do for us, but through curiosity, openness, listening, and observation. So often we are quick to look for easy solutions to problems, but conservation mycology is not a simple problem. This is a complex situation because of their inherent interconnected nature and the many other characteristics I shared before. Fungi show that what is true for a lot of different fungi show what is true for a lot of different organisms, that we are interconnected in multitudes. It's easy sometimes for us to jump to action and ask, what can we do without first listening? Listening and observing fungi instead of jumping to just doing. After listening, we can then expand on just being aware. We can go from noticing, being aware to actively sharing our delights and love of fungi with one another. For example, learning how to center celebration and conviviality of fungi rather than fear-driven research, scarcity-focused approaches. How do we also unlearn cultural mycophobia of fungi being only associated with death, disease, and decay? One great example I've experienced is a lot of the mushroom gatherings out there. Telluride Mushroom Festival here out in the Colorado woods, playing with fungi and celebrating with each other. And I really love their mushroom parade that they have at the end of their festival, where my friends and I dressed up as entomogenous insect-loving fungi, like Bavaria, the fly death fungus, and cordyceps, and Massospora. Another example of the future of fungi can be holding spaces for grief and sharing our love of fungi in the environment. As many know the joy of encountering a mushroom, sharing this joy is not only infectious, but can help others care and notice fungi. We can create spaces where we can share delight. I've noticed in the spaces that I've done this in, where I share delight, it just keeps on coming. Just noticing more and more delight, more joy, and it's a great way to bond, especially during heavy times when there's not a lot that's easy to talk about. We can continue to expand on just being aware and celebrating fungi to learn how to be stewards, how to get our hands dirty. So stewardship of habitats rather than management or letting nature do their thing on their own, like traditional old styles of conservation. So how do we create conditions for fungi to grow successfully and flourish? So perhaps more observation focus and active participation, less controlling, rather than, focusing on conditions for success. We use the word stewardship as it contains an ethics of responsibility rather than land management for entrepreneurial profit and efficiency. Along the way, we can shift our conservation paradigms to be more collective focused, abundance rather than scarcity. What we focus on, we reproduce. If we only seek scarcity, we'll only find scarcity. There is an abundance of fungi living today. Why not conserve the abundance we have instead of waiting for it to become scarce? Another shift in conservation paradigms is to be more community focused, focusing on collective functions of fungi rather than individual sporing bodies or a species focused. This helps us address the issues of interconnectedness, shifting paradigms, invisibility, scale, and diversity I previously had mentioned. I really think these, this like conservation of abundance mentality and collective functions focus is a game changer and really helpful. And how does a collective group of fungi and plants and microbes work together in an ecosystem? What do they uniquely offer? So you'll notice that monitoring is just a few of the questions that make up our current conservation research agenda of conservation mycology. That is not to say in any way that is insignificant, but just one of the many items of inquiry that conservation mycology is interested in. There are indeed noble monitoring efforts afoot, like the CA Fundus project and Fundus's regional rare fungi challenges, which are very successful, as well as the natural heritage programs in Pennsylvania and Oregon. Yet one of the reasons California the CA Fundus project received funding was because their project was committed to researching associations and relationships with other species of plants and animals and fungi. Efforts are becoming more and more habitat and relationally focused. And we know fungi are well suited for helping us think more relationally in their entangled, interconnected ways of living. Yeah, by recognizing the valuable labor and work fungi do in tandem with other species for ecosystems is one of the many ways fungi can help us shift our conservation paradigms. These concepts of abundance and functional collectives are talked about at length in one of our favorite papers we reviewed in the committee, and it is big inspiration for my talk by Elizabeth Barron, The Conservation of Abundance, where she shadowed a mycology lab and reviewed the conservation mycology research and came up with a focus of conservation research that's focused on abundance and more of these functional collectives and just collective focus, community focused. If you're interested in learning more, I suggest reading it. I've expanded on it quite a bit in this talk. After we have some ideas on how to steward or notice fungi systematically, we can look for funding. So funding and organizing approaches. So we can think of come up with ways that are both bottom up and top down. So bottom up organizing to get funding. We could do community organizing, grassroots organizing, where we're going out and collecting species and community lists to propose to conservation to government agencies like the natural heritage programs and granting agencies to fund our work. Or we could do how we did it with the CA Fundus project at top down, advocating for governments to include fungi in their conservation frameworks and 30 by 30 initiatives. Um, yeah, and it'll take both approaches and everything in between, as there's no one size fit all approach to get funding. In general, is to our advantage to help conservation be reimagined to include fungi and fungal habitats wherever conservation is happening. All right, so here I think is a fun part where I will talk about how y'all can get involved, how you can be a steward, and what are some best practices for fungi and looking for mushrooms, how you and your community and fellow NAMA members can get involved. So as a member of NAMA, we have a responsibility to model what best practices are in conservation and stewardship for fungi and their habitats they live in. So what can we do for fungi, and how can we practice looking for mushrooms responsibly? The following relational approach to conservation that I will be talking about is deeply inspired by Robin Wall Kimmerer's Honorable Harvest, which she describes as an unwritten set of indigenous principles and practices that govern the exchange of life for life. They are rules of sorts that govern our taking, shape our relationships with the natural world, and rein in our tendency to consume. That the world might be as rich for the seventh generation as it is for our own. The first off is the know the ways of the one who take care of you so that you may take care of them. Introduce yourself. Be accountable as the one who comes asking for life. Ask permission before taking. Abide by the answer. Never take the first, never take the last. Take only what you need. Take only that which is given. Never take more than half. Leave some for others. Harvest in a way that minimizes harm. Use the harvest respectfully. Never waste what you have taken. Share. Give thanks for what you have been given. Give a gift in reciprocity for what you have taken. Sustain the ones who sustain you, and the earth will last forever. These are all from Romuald Kimmerer's Braiding Sweetgrass. I highly recommend it to read the book as a whole. Additionally, the practices that I'll go on to share have been influenced from my professional work with the Fungal Diversity Survey and my involvement with the NAMA Conservation Stewardship Committee. So through the committee, we've done our literature reviews, and we are also actively gathering best practices from conservation-focused organizations and our 90+ affiliated societies. Our committee is still working on publishing a set of guidelines we collectively agree upon and think should be shared with the larger public. As I had mentioned earlier in this talk, there are only a few published peer-reviewed papers, research papers on best practices. So the following are best practices that are a work in progress that our committee has largely put together, and I've adapted a lot of them. My hope is that you will use these guidelines as you build relationships with the more than human world, helping fungi thrive and reduce any intentional harm. So the first few I'll go on, I'll just share it. So before you go into the field, avoid gathering in heavily populated areas to prevent overharvesting. Explore local concerns and issues related to fungi and their environment. Be aware of the sensitivities in your environment. Each environment has their own unique set of challenges. Learn whether landowners' permission is needed to visit or gather. Pack an extra bag to collect any waste you come across on your walks. In the field, harvest in a way that minimizes harm can look like leaving decomposing trees the way you found them. Cover any exposed soil with leaf litter or fill holes left by collecting mushrooms to leave the soil as you found it. Minimize disturbing the soil in the leaf litter or duff. Give a gift in reciprocity for what you have taken can look like leaving behind some water. Water is something I almost always have with me, and it's something I can share. Water is life, as the adage goes. Walk in wild areas with care. So research has shown trampling and going off trail can cause damage to fungal habitats. So use the trails. When we're walking off trail, walk lightly, carefully stepping between plants without damaging them. You can use trails made by deer and other critters to further lessen the impact. Staying close to managed trails will also help just in general reduce your impact. Take mature mushrooms. This helps mushrooms by giving them enough time to spread their spores. Allow perennial polypores to grow continuously from year to year by leaving them undisturbed. So, what can you do to help conserve fungi and their habitats? A bit of this is actually on our website, on our resources page. But first off, you can educate yourself. So learn with others. Learning with others is so much fun compared to learning on your own. So join a local fungi education community, like exploring the mycoverse, or a book club, or one of the 90 plus NAM affiliated organizations. If you want to get deep into it, start your own community research group where you read published literature or fungi books. Google Scholar is a helpful resource for finding accessible papers. And then for advocacy and activism, join our NAMA Conservation and Stewardship Committee. We have monthly meetings, and our next one is next week, Wednesday, January 22nd at 5 p.m. Pacific or 8 p.m. Eastern Time. If you're interested, email us at conservation@namyco.org. And I can put that in the chat after this too, if you're interested in joining. And our email should be on the website as well. We're interested in hearing what our members are doing for conservation research and stewardship practices. We'll be sending out, so keep an eye out. We'll be sending out a survey to learn more about what our membership is doing and how NAMA can help you in those efforts. So please keep an eye out for that and complete our survey. And keep an eye out for future scholarships and apply to those. We recently received word we have some funding for scholarships or a scholarship, so keep an eye out for an application in the near future. Yeah, we're actively looking for members who are interested in showing up once a month. We have so many ideas and things we want to work on, but we're limited by the number of people we have. And we also need a diversity of voices to accomplish what we want to accomplish. And yeah, last but not least, check our resources page. Further for advocacy and activism, advocate for fungal awareness and inclusion in existing conservation frameworks. Include fungi in 30 by 30 goals. Volunteer with native plant restoration projects and non-native plant management. Promote and support initiatives that enhance biodiversity in soil and prevent soil erosion. And another way you can be an advocate for fungi is by being inclusive of your fungi, being inclusive fungi in your language. So describe fungi on their own terms and learn fungal specific language. So refrain from using language from botany or other fields. Like I said previously, sporing body instead of fruiting body, mycelia instead of vegetative body. Adopt the term funga and encourage others to do it as well. And if you didn't know, funga, the fungi of a specific region, habitat, or geological period, it is a term that is equivalent to the concepts of fauna and flora. Giuliano Furci, the founder of the Fungi Foundation, came up with this term to incorporate fungi into existing conservation frameworks. This is part of a call for organizations to adopt the usage of the word funga when describing macroscopic life, such as descriptions of environments wherever fauna and flora are mentioned to describe the ecology of a place. The call is known as the Fauna, Flora, and Fungi Initiative, the FFF, the 3F Initiative, collaborative endeavor between the Fungi Foundation, NYU's Earth Advocacy Clinic, and Merlin Sheldrake that seeks to elevate the status of fungi within the realm of conservation and environmental protection. I really love it. We've adopted it recently at Fundus into a really beautiful design by Tay Bright, Califunga, as in Califunga, dude. I love that. We can do, there's state level advocacy. So contact your state's natural heritage program, which is often housed in the Department of Natural Resources. You can find yours on natureserve.org. Write to your state legislators expressing your support for fungal conservation initiatives. If your state doesn't have an official state mushroom or state lichen, start a campaign and include some candidate species that lend themselves to talking about conservation. Recently, we just got our California state mushroom. We had a state lichen well before a state mushroom. And our state mushroom now is the California golden chanterelle, Cantharellus californicus. Oh, and there's the definition of fungi. And other ways you can get involved is by becoming a community scientist, by documenting your observations of fungi. Really important for gathering accurate data. Our conservation strategies depend on this to know what fungal species exist and where. So you can do this by creating an inventory of your fungal encounters using apps like iNaturalist or Mushroom Observer. Monitoring fungi can help with identifying the spread of potential invasive fungi, which we don't still know much about yet, but is definitely occurring. And you can support fungi collections at fungariums, the collections of natural history collections, and organizations doing this work like the Fungal Diversity Survey. And you yourself can start a fungarium, which is different from an herbarium that is dedicated collection to plants. So a fungarium is a dried fungi specimens like a herbarium, but for fungi. And their primary function is to provide a well-organized and classified repository, enabling scientists and scholars to easily access these samples of fungi and um, do their research. They serve as a very important record for the future. Fungariums can be created by individuals and housed in people's homes, or they can be created by institutions and housed in botanic gardens and universities. The largest fungarium in the world is hosted overseas in London at the Kew Botanic Gardens. In North America, there is a growing need for additional fungariums to be created as mycological research grows. At the time of this writing, here locally, UCLA is just establishing their own fungarium with the help of the California Fungal Diversity Survey project. Because we've been running out of space, we've collected thousands upon thousands of fungi. And so if you're interested in becoming a fungi community scientist, there are some easy to find tools and equipment you can use to make this science accessible for you. Here's an example of a field toolkit a fungi collector community scientists can use. So this is an example of my own I most often use. My trusty hand lens to view the tiny fungi, a 365 UV light. So many fungi have characteristic fluorescence under UV light that help you can identify fungi very easily by their fluorescence. And we have tackle boxes for protection and organization of specimens, wax paper bags for larger specimens. We've got a mushroom knife for harvesting and gathering, KOH 5% for diagnostic staining, field data slips, which you can find on the Fungal Diversity Survey. It's a website, water-resistant notepad and pencil. So just something, you need something to take your notes on in your field. It's really easy to get too excited and forget to take a bunch of notes when you meet a mushroom. I've learned from experience. And then a macro lens for your phone or your camera, if your camera has a macro lens. So yeah, cameras with photo stacking capabilities are really great to help show the depth of field. It's a bonus, like the Olympus TG series camera. Not sponsored by them, but I really like that camera. It's been incredible. Many of the photos in this presentation have been taken with that camera. And then tweezers, those are really helpful to manipulate small mushrooms and collect them. And one of my favorites I recently learned from my friend Yannicka, who visited a conservation reserve outside of Amsterdam, is a telescoping mirror. So you can look at the underside of mushrooms without having to pick them. Here's an example of using it up close. Here, didn't have to pick the mushroom. I set the mirror underneath, and I can see that it has purple gills, knowing it's a bluet. Otherwise, just another little brown mushroom. And you never know what you'll find under those mushrooms. So what else can you do to help conserve fungi in their habitats? So in practice, Juliana Furci of the Fungi Foundation says, let things rot. So leaving decomposing trees and other woody debris to support wood rotting fungi. A lot of fungi only grow on certain trees, so big logs, some I've noticed a lot of crusts only grow on big trees, not twigs. Yeah. Other ways is to leave the leaves, leave a layer of leaves in your garden beds under the trees or less used areas of your yard. As I said, many fungi grow exclusively on larger woody debris, such as stumps, dead standing trees, and falling logs. Um, through gardening, oop, through gardening and yard maintenance, you can support fungi by minimizing or eliminating the use of fertilizers that prevent mycorrhizal symbiosis. Uh, you can minimize or discontinue tilling as it disrupts fungal networks. And that kind of brings up the soil, breaks those connections. Um, and learning ways to remove water sterilizing agents like chloramine from your water that you use when watering can kill microbes like fungi. Chloramine is a popular chemical found in water in Los Angeles. And here's a really cool example of a cross fungus I love. Um, Other practices you can help fungi by avoiding leaving soil exposed. This is a huge one. Exposed, bare soil lacks protection from heat and does not retain the moisture necessary for our underground fungi. Preserve large and older trees and ensure a diversity age range among the trees in your forests. And be cautious of when cultivating non-native species of fungi. So avoiding discarding spent spawn in areas potentially suitable for a fungus to spread into the environment. Um, and support your local mushroom growers. I think this is another way fungi can help us rethink conservation. So conservation is simply how we interact with our land. You know, buying local mushrooms and food has substantial benefits to not only us, but the environment. Most farmers markets have at least one stand selling farm fresh mushrooms, at least here in Southern California. Not only are the locally farmed mushrooms tastier when they are fresh, but they are also healthier. Um, Buying from local mushroom growers means using less plastics and fossil fuels for transportation and storage. Buying local also means supporting local economies, regenerative agriculture, and more socially responsible agriculture. Yet these are all things you need to ask when you go out. You know, it's no different when from other cultivated foods you can purchase. So check with your local mushroom growers on how they grow their mushrooms. Um, are they growing out their fungi in a whole foods diet? Are they grown sustainably? What do they mean by sustainable? Are they grown on organic products? Are the farmers practicing circular regenerative agriculture? Are they grown socially responsibly, such as paying workers a living wage and intentionally using less plastic and water? This way, mushrooms can be a very sustainable calorie and significantly use less amount of water, waste, and plastics and greenhouse emissions that growing them requires, especially compared to animal meat. All right. Um, in conclusion, while all these ways are ways to get involved, going back to the basics of rooting our actions and our deep love of fungi is a proven approach to all kinds of conservation. If you haven't already, reflect on how you can share your love of fungi and the environment with others. Is it through art, a book club, scientific observation, teaching kids, leading walks at your local park, or is it gardening? How can you get your hands dirty to love the world back to wholeness together? I want to end with my Eden Canyon story. I think it's a really beautiful story of falling in love and joy and conviviality, and how we will love this space back together after these fires. Eaton Canyon is in Pasadena, California, is the introduction of fungi for many urban folks. Eaton Canyon is where I led some of my first walks with mushrooms, with kids in schools and the local California Native Plant Society, where, as I mentioned, I met my partner. It's where I've learned how our native plants support multitudes of fungi. My exploring the mycoverse community, and I have led extensive fungal surveys in these areas for both my SoCal Funga project and the CA Fundus project I work on. Beneath the majestic trees in the canyon, we've encountered a thrilling variety of mushrooms, including black jelly roll fungi, Exidia glandulosa, covering the trees so thick they were dripping from the trees after the significant rains we've received the past 2 years. Here is a picture of the stinky bleach and talomas that we've encountered under our oak trees. And then we have the milky yellow staining Lacterius xanthogalactus. So interesting. It starts out white and then it turns yellow. The latex does. Beautiful. And then we have the amorphous caps with white ribs of elfin saddles, Helvella dryophylla, many who are covered in white Helvella eater mold, as such as the one on the right, Hypomyces cervinin genus, as well as many other tiny fungi, such as the smoked oysterling Recipionatus applicatus, which I will share a photo in a moment. But one of my favorite fungal encounters on those walks was an undescribed species of Xylaria, who I call phantom fingers, as they have white finger-like sporing bodies that branch upwards from dead wood, looking very ghostly. Under the UV light on the right and the bottom left, the sporing bodies fluoresce a fantastic turquoise with green dots that highlight the mushroom sweating of metabolites, known as guttation. While mushrooms cover the walls of Eaton Canyon, I learned how much microclimates can affect fungi. Inside each of these canyons, the microclimates vary, providing differing habitats that host unique fungi specific to that canyon's conditions. Another example of this is coming across an undescribed species of stalked puffballs known as Telostoma. They are particularly semi-arid loving mushrooms that grow in sand. It's interesting because the canyons are some of the wettest and most humid parts of the Los Angeles area, with water year-round. In fact, these year-round water resources, indigenous people have lived in these canyons for thousands of years. The unique biocultural diversity these canyons hold deserves to be protected from future development and continued to be stewarded. Efforts have been made to attempt to develop these areas, but with the help of the community, these efforts have been stopped. There's a local Eaton Canyon Center that is filled with its own resident ecologist, herbarium, restoration stewards, and a team of volunteer docents, some who are a part of exploring the mycoverse, who educate park goers about the native plants and fungi, who I work with and recently have given an ecological focus workshop on. And that's the smoked oysterlings on the left I love so much. But thank you everyone for coming out tonight and joining us this evening. Please reach out. I would love to hear your feedback on this talk. This is my first time giving it as a whole single talk. Typically, my talks are more discussion based, and I just share parts of the material that I've shared tonight. And hopefully, we still have some time for Q&A where I can take some questions.
Hey, Aaron, thank you very much for that. Much appreciated. Everyone, please feel free to give your thanks in the chat. We don't have too many questions. In fact, we have one right now, though usually people come up with more questions as they come along. But the presentation went a little long, and so we don't have too much time for the for Q&A anyway. So we'll get to that question. Oh, there's another question that just popped in. So here we go. So the first one is from Veronica. What were your methods for coming up with the organized questions from the conservation mycology tries to answer and the themes of concern that you had listed in your presentation?
Yeah, that comes directly from that paper, the recognition of conservation mycology as as a discipline. So that is a list of conservation mycologists that have been around for a long time that I was simply putting in my own words.
Okay. We have a question here. Someone's looking for basically an email that they should reach out to in order to join this conservation committee of NAMAs.
Yeah, you can reach out to my email listed here, and I will put you in touch. And I could maybe, yeah, we also have myco.org. All right, I put it in the chat too.
There you go. Conservation at NAMI or YCO.org. So namico.org. Conservation at. Is anyone able to join a Fundus project?
Yeah, if you're you can check it, especially on iNaturalist, really easy to use. Your observations, if it's in a certain area, will automatically be collected or submitted to those. And you can reach out to your local mycological societies. They will have their own projects that they run. So sometimes they're for a specific time frame or a specific uh, area, like we do at different botanic gardens or gardens I focus on, I create projects for. But yeah, check out fundus.org. As I see, a lot of people are advocating for in the chat. I love
No
that.
kidding. It's been all about Fundus in this chat or presentation long. Following up on the Fundus question. So there's a question, I think it was posted in the chat earlier. It's again being written here by Myco Habitat, who asked, is there going to be a post-fire fungus survey to document all these pyrophilic fungi in the canyon? I assume it's a little early days to have that conversation, but presumably something's going to be happening, I assume.
You bet. It will be a bit tricky because a lot of these areas are highly toxic and we're having to figure out navigate those areas because this is an urban fire. But yeah, there are already discussions with the Eaton folks that they're really excited to see uh, how this fares because actually there's a good chunk of the oaks that were not burned on the western slopes I heard today. So yeah, good. And that's actually where a lot of fungi are. But yeah.
So there's a kind of a follow-on question to that, which is for mycoremediation efforts in response to these fires. Do you know of anything along those lines?
Yeah, it's too soon yet to organize those, but I know there are folks who are very interested. I've been a part of a mycorradiation project by Daniel Stevenson in the area. But yeah, there are organizations like Co-Renewal who specialize in fire restoration, post-fire. I know my colleague Tate Bright is is well versed in this. Um, but yeah, I'm excited to see those. So stay tuned. We'll definitely be working on this in the future.
So I suppose if people would like to hear any updates on that front, maybe they should reach out to the Conservation Committee. If you guys do find out anything in time, I'm sure you'll pass along the information.
Absolutely. Yeah.
So we do have one last question written by Ken Sanderson. A lot of discussion on the conservation seems very focused on what we can do, i.e., let things rot, picking practices. But what about the larger picture stuff like landscape conservation?
Landscape conservation. I think that is where we'd really love to go is to create some conservation reserves, like there are in other countries, like I mentioned in the Netherlands. There are very few, but they are out there. But yeah, in general, we're working large, big and small, just to get to know fungi. But yeah, I think including just getting some more research, some more funding for research so we can ask those bigger questions, getting fungi included in the wherever there are, fauna and flora mentioned, include fungi. Because yeah, we I can't stress enough, but we don't have very much funding. I mean, I'm working 5 different jobs to support myself to do this work. So yeah.
Yes, indeed. We do need to get it more top of mind, I think, for the average person. And then from there, finances will flow and we'll be able to do a lot more. Money speaks volumes, unfortunately. So I definitely agree with that. I don't have any other questions for you at this time, Aaron. Is there any last thoughts you want to share with anyone?
No, just a lot of gratitude. Thank you for coming out and your interest in the fungi. And I will be really curious to hear what y'all do with this information. So please reach out. I'd really appreciate that.
You had mentioned that you have a book coming out. Do you have a date for that yet?
I don't yet, but I'm very excited. That's yeah.
Or a title, Veronica says.
Yeah, it's
Title
still
yet. All
process.
right.
So
Soon to
yeah.
be coming out. We'll
Yes.
keep an eye out for that. Aaron, again, thank you so much for taking the time to do this. I know your life is kind of up in the air right now, but we really appreciate it. There's a lot of great information that you shared. I'm sure everyone here really enjoyed it, and you're getting a whole bunch of thanks in the chat section. I'll leave it open for a minute longer, but yeah, I just wanted to say thank you so much once again, and it's been a pleasure hosting you.
Thank you, Robert. Thank you, everyone.